Hello, ma'am, and all my dear fellow mates. Myself, Anuradha Chadon from MSc Biotechnology 3rd semester. And today, our group is going to present the review paper entitled Feasibility of Acrylic Acid Production by Fermentation. Let's know what is acrylic acid. IUPAC name of acrylic acid is propanoic acid. It's an organic compound having 3 carbons. It's the most simplest unsaturated carboxylic acid. The major utilization of acrylic acid is in polymeric flocculants, dispersants, coatings, paints, adhesive, and binders for leather, paper, and textile. The pKa value of acrylic acid is 4.26, hence, it is mostly deprotonated at neutral pH. Acrylic acid is conventionally produced from petrochemicals. In single-step process, partial oxidation of propene results in the formation of acrylic acid. The yield is 50%, but the demerit is large amount of waste is produced along with. Hence, two-step production is preferred, in which acrylic acid is produced from propylene via acrolein, in which 90% of the yield is produced. But the thing is, both these methods have non-renewable petrochemical sources. Hence, they emit large amount of carbon dioxide, and these sources will become scarce and expensive in future. Worldwide, supply of propylene is diminishing. Plus, it is not eco-friendly. Therefore, our purpose is to produce acrylic acid from renewable carbon sources in an economically preferential manner. Metabolic engineering approach will aid in the development of an efficient host microorganism, its insertion into a host organism. The pathway will be designed in such a way to have a balanced mass, feasible redox reaction, biochemistry, thermodynamics, and will theoretically give maximum yield of acrylate on glucose without any aeration or extra energy requirement in the form of electron acceptors. Candidate pathways which can be considered are through beta-alanine, methyl citrate, or methyl malonate coenzyme A. Hypothetical conceptualization of acrylic acid production involves target production of 1 lakh ton per fermentation, easy product recovery, efficient host organism which is resistant to acrylate toxicity, feasible biochemistry, thermodynamically stable reaction, and a cost effective method. Acrylate shows both positive and negative results in vitro genotoxicity test. Acute toxicity in fish is due to presence of double bond which makes it reactive, and when these acrylic acids are ingested by fishes, double bond reacts with glutathione which results in the depletion of GSH, which in turn results in the cell death of fishes. Acrylic acid is hydrophobic and acidic. Hydrophobic means it is water resistant. Acidic means it has tendency to lose electron, which makes it extremely reactive, which results in narcosis. Narcosis means general depression in biological activity due to presence of toxic elements. These toxic elements we can say in terms of acrylic acid is its hydrophobicity and acidic nature. On the other hand, Clostridium propionicum is capable of rapidly metabolizing acrylate in a 35 gram per liter of acrylate solution, although it couldn't grow in this solution. Hence, we can say it gives both positive and negative toxicity genotoxicity test. So, how can we increase microbial resistance to increase acrylic acid production via fermentation? It can be done in two ways, either by selective pressure or by genomic shuffling. What is selective pressure? Selection of resistant bacteria that can survive in extremely high titers of acrylate. Genomic shuffling is recombination of genes to enhance the strain. Despite the high reactivity of acrylate due to double bonds and toxicity due to high titers, still fermentation process.
My name is Vanshita Rawal and I will be starting from the role of acrylate in microbial metabolism. So our goal is to produce acrylate from renewable energy sources in spite of non-renewable energy sources. DMSP एक ऑस्मोलाइट है जो कि मराइन ऑर्गेनिजम्स में प्रेजेंट होता है. अब DMSP is also an energy source for certain marine bacteria. This bacteria will degrade DMSP into dimethyl sulfide and acrylate. Now we can see anaerobic organisms में this is like further conversions होते हैं जिससे हमें acrylate मिलता है. But in aerobic bacteria हमें एक्स्ट्रा सेलुलर एक्राइलेज देना पड़ता है ताकि DMSP acrylate रिलीज करे. Now we can see An enzyme acryl CoA is come into a role, that is an intermediate of 3HP cycle. साइकिल से हमें 3HPCO एंजाइम मिलता है जो कि डिहाइड्रेट होता है एक्राइल कोए में, और एक्राइल कोए रिड्यूस होता है इनटू प्रोपेनाइल कोए। बट सर्टन बैक्टीरियाज ऐसे देखे गए हैं जिसमें ट्राई फंक्शनल एंजाइम रहते हैं जो कि सारा ये प्रोसेस को खुद ही कर देते हैं यूजिंग एटीपी। एक्राइल कोए जो है दैट इज़ एन इंटरमीडिएट इन द फर्मेंटेशन ऑफ लैक्टेट. So, in this fermentation, lactate is converted into two sections, like 2/3 part of it is reduced to propanoid and 1/3 part is oxidized to acetate and carbon dioxide. So, the author has suggested that instead of using lactate, we can also use alanine. Now, moving forward to directing metabolic pathway to acrylate. So here is the figure. We can see the several routes that are reaching to the formation of acrylate. Pathways are linked. So the points we can see here is like propanoid CoA do not require propanoid as an intermediate, whereas like the lactate to acrylate is a direct route for the production of acrylate. Thus, we can block other pathways in order to for the more production of acrylic. But in this, we can see certain problems occurs. So the problems like the lactate do not totally convert into that acrylate. Some part of it is converted into acetate and carbon dioxide. And here we see that we cannot stop that production of acetate, as acetate is also an important component for the growth and maintenance. While other problems like the acetate and lactate ratio, the equilibrium has to be maintained, like it should be a high in order to get the high yield of acrylate. And other problems are like many routes are rely on dehydration of 3HP, but it is seen that अगर हम dehydration करेंगे पूरी प्रोसेस में जस्ट फॉर दैट द सेक ऑफ कि हम 3HP अगर डिहाइड्रेशन होगा तो वो आगे प्रोसीड करेगा कन्वर्जन्स एंड like we can get acrylate, but dehydration is unfavorable for like fermentation. So this problem is also a barrier in the production of acrylate and many more. So like there is, so far it is not clear how to obtain a high yield of acrylate on sugar.
Hello, I'm Nitisha and I'm presenting selection of most attractive routes and export. Firstly, minimization of cost of sugar to acrylic acid formation is done by using single bicatalyst for the whole pathway. There are many indirect and direct routes for the acrylic acid formation. In the indirect route, many intermediates are formed and by their chemocatalysis and bicatalysis, acrylate is formed and finally recovery of acrylic acid is formed, which is not a feasible process because it is very complicated. And in the direct conversion there is a sugar to acrylate formed directly and it is very feasible process. These are various metabolic pathways in which various intermediates are formed. If we want economically competitive fermentation process, then molar yield of acrylate should be almost quantitative. That is, there is a production of 0.8 kg acrylate in 1 kg of glucose. If we want to overcome reaction equilibrium, then the driving force for the reaction should be large and there should be no competitive pathways to the acrylate formation. During the conversion of sugars to pyruvate, 1 ATP is generated. By this, no additional pathways are required. If there is additional pathways, they consume glucose, require aeration and produce other fermentative products which are undesired. Furthermore, conversion of pyruvate to acrylate require no ATP and they neither use generated ATP. To form actual beta alanyl CoA require 1 ATP which they get when acrylate is released from CoA. To enhance driving force, Reaction is catalyzed by a transferase. There are two potential pathways from pyruvate to acrylate via propanol-CoA. First is reverse methyl citrate pathway and second methyl malonate-CoA pathway. In reversed methyl citrate pathway, there is a alpha oxidation of propanoid to pyruvate in S. cerevisiae and many other organisms. There is also involves oxidation of succinate to fumarate involved NAD as a redox cofactor, and if we reverse this cycle there is a issue, that is NAD is close to cytochrome B and this is also used for the oxidation of propanoyl CoA to acrylyl CoA, but their driving force is low. To enhance their driving force we have to done hydrolysis of acrylyl CoA. And second is methyl malonate CoA pathway used to convert pyruvate to propanoyl CoA. In this, fumarate is reduced to succinate and propionibacteria generates 2 ATP. These 2 ATPs are neither used for the formation of acrylate nor to drive the oxidation. Beside this, the oxidation potential of fumarate and succinate is used. A study is there in the comparison between enzymes of Vollinella succinate that carry two redox reactions. If we want to decide the pathway, there is analysis of metabolic flux required. First, analysis of consequences of blocking the pathway of potential side products, and second, whether the host organisms has enzymatic activity to change the propanoyl CoA to propionate because they causes disproportion. Probably, an efficient metabolic pathway will require the absence of hydrolysis of propanoyl CoA when there is a acrylyl CoA hydrolysis. An enzyme with such specificity might have to be found or developed with respect to redox enzymes for fumarate, succinate, or acrylate propanoid. Many other issues will have to be studies like cofactor involve the regulation of metabolic pathway and the product export, etc. Both acrylic acid show an antimicrobial effect have to be exported efficiently by the cell. The microorganisms will have to maintain its electrochemical gradient across its cytoplasmic membrane. For the feasible type of export, some thermodynamic constraints occur. If associated protein is actively exported, there is an acrylate anion exporter used having pH7 and internal to external concentration ratio of about 0.01. If the undissociated acid is exported, then ATP-driven transport proteins belonging to the ATP binding cassette ABC transporter family are used having lower concentration ratio. All ATP from acrylic acid production are consumed by export, thus there is no ATP left for growth and maintenance, so there is need to addition of some sugars and this associated sugar and aeration cost economically unacceptable. The process is anaerobic and selection and designing assuming that microorganisms might become available that enables the production of 50 gram per liter acrylate, which is a good amount.
Thank you, Nitisha. So, hello everyone, I'm Muskan Chandel. Let's put forward our discussion further. Now, we are discussing how acrylic acid produced and how that process gets selected and designed. So, almost 12% of consumed sucrose as a sugar source used for cell growth. And here we use continuous fermenter where anaerobic and aseptic condition applies so that the relatively modest amount of cell produced and having high volumetric productivity. Then transfer to disk-stack centrifuge whose principle is to remove solid from liquid or separate the two liquid phases from each other. And here we chosen it for the cell removal as waste. Now the most significant step is the recovery of acrylic acid. There are three operations. First, stimulating moving bead ion exchange chromatography. Second, liquid-liquid extraction and ammonia stripping with liquid-liquid extraction. Here, liquid-liquid extraction is the key purification process, but in this paper, SMB, that is stimulating moving bead ion exchange chromatography, mainly focus, and here in SMB, it forms the aqueous acrylic acid stream from which water should be removed. And for this problem, preloaded carbon dioxide gas is used to lower the pH and organic phase is used. To dissolve the sufficient amount of carbon dioxide, relatively high pressure is used, mainly 16 bar. Carbon dioxide lower the pH and convert the acrylate salt into acrylic acid. Product having organic salt shifted to distillation column where water removed from organic phase. The left product then sent to second distillation column where acrylic acid purified. The remaining again sent to this extraction to purify the organic phase from the impurities. Now next is economy. The figure shown is according to Senate. Here, the fixed capital investment is calculated with overall direct and indirect cost of worth 64 million euros, and with the addition of working capital and startup, total capital investment worth of 86 million euros. According to this figure, total manufacturing costs were 96 million euro per year. Here, sucrose dominate all over the raw material. If we see the percentages, sucrose is 64%, sodium carbonate is 27%, and ammonium and other nutrients are only 9%. In this figure, for 15-year economic lifetime, cumulative net present value is 220 million euros. The net of return is 17% and internal rate of return is 34%. Process economic acceptable only if the acrylic acid selling price reduced by 20%. Last is conclusion. In whole paper, the center of attraction is metabolic pathway which converts sugar to acrylic acid. The effective expression of any pathway in host organism will require extensive engineering and might be ever require some yet unknown enzyme activities and export protein. Pathway need to be evaluated in more detail to show the microorganisms would generate ATP. Economic evaluation indicate that there is a clear incentive for the development of required microbial host, in particular considering environmental sustainability of design process. So that's all from our group. We hope you like it. Thank you so much.